Hello and welcome back to Day 2 of Cell and Gene Therapy 2024. I'm Heather James, Conference Producer at Technology Networks, and I'm here to introduce our first talk of Day 2, Unlocking the Potential of Memory T Cells, Harnessing Commensal Bacteria-Derived Peptides for Cancer Immunotherapy. Unfortunately, I do need to pass on apologies from Pierre, who cannot attend due to unforeseen circumstances. However, I am thrilled to welcome in his stead Dr. Laurent Chene. Laurent is the head of drug discovery at Enterome, a clinical stage biopharmaceutical company developing breakthrough immunomodulatory drugs for cancer treatments. He obtained his PhD from the Pasteur Institute working on HIV before moving into the industry. Laurent joined Enterome in 2015 with 15 years' experience in target identification, screening, and drug discovery. A warm welcome to you, Laurent. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And without further ado, I will now hand over to Laurent to begin his presentation.
Thank you, Heather. So thank you first for the invitation. So we are very pleased to present our platform at Enterome, mainly associated with discovery of peptide, Commensal Bacteria-Derived Peptides for cancer immunotherapy. So first, who we are. So Enterome is a clinical stage company which is developing a new generation of immunomodulatory drugs. So we are 35 people in the lab and 30 people in the office. Currently, we have already 4 programs that are in clinical phase 2. Until the beginning of the company, 10 years ago, we have been able to set up a large number of strategic partnerships. For example, we have get more than 100 million in non-dilutive funding and a lot of money in project milestones. We have been able to set up collaboration with diverse pharmaceutical industry, including Pfizer, AbbVie, Takeda, J&J, and BMS. And we have also leads by international venture and corporate investors, namely Nestlé, for example, BMS, but also Venture, Science, Olmos, and many others. So basically what we are doing on our platform is that we are working on what we know from the human gut microbiome. And at the beginning of the company, we were able to go very, very deep in the investigation in all of what we found in the human gut. So we were able to sequence more than 26,000 human gut microbiome samples, and we have been able from this to get, let's say, to characterize around more than 20 million of unique bacterial proteins. So this is the starting point for us to work on compounds or antigens that will drive immune response or modulation of the physiology of the human gut and the human body. So why? So this is linked to the unique ability of the bacterial protein and the bacteria themselves to elicit a crosstalk with the human body. So at the level of the gut-human barrier, you have a lot of interaction between the bacteria that will drive immune response. So you know that the immune response is mainly primed by the bacteria that we have within the gut, but there is a lot of compounds, also bacterial compounds, that can modulate the suppression of different cytokines and hormones within the gut. So basically, we are working on that. We are not working specifically on gut, so we are not developing drugs from bugs. We are really looking at different antigens, different compounds that are associated with biological activity directly on the immune system and on the immune gut barrier homeostasis. So basically, from this start, from this knowledge that we have been able to develop several programs. So one is linked to oncomimics, and this is the program that we will discuss during this meeting. We have also programs on endomimics and programs on allermimics. So oncomimics, what is it? You will see in the presentation. This is bacterial antigens that will be able to wake up immune response against tumor-associated antigens. Endomimic, what is it? So this is bacterial compound that will be able to induce, for example, pro-inflammatory, anti-inflammatory response within the gut. So for example, we have compounds that are able to induce IL-10 secretion within the gut, or that can modify secretion of some peptide associated with satiety or metabolism. And we have also programs that are co-developed with Nestlé Health Science on allergenic. So here, the objective of this third project is more to identify compounds that will be able to blunt immune response associated with allergy. So during this presentation, I will mainly talk, as I said, on Oncommunic platform. So what is this Oncommunic platform? This is a way to utilize non-self tumor-associated antigen to activate and expand T cells for the targeted destruction of cancer cells. So on which things is it based? So first, we know that T cells are the most effective way to kill cancer cells. So a lot of companies are working in different tools, therapeutic tools to try to activate T cells in a way that they will be able to kill tumor cells. For example, you know, CAR T cells, but you know, say, TCR engineered T cells. So this is very fine. We know that T cells are the most effective way to kill cancer cells, but unfortunately, there is a lot of factors that limit their use in a large population. There is some side effect and difficulty in sustainability of the immune response associated with this method. So what we have done at Enterome is that we have tried to check all the limitations of the current therapeutics that exist and that are using T cells activation for inducing anti-tumor immune response. So first, we are overcoming self-tolerance to tumor cells by using cross-reactivity and inducing immune cell response against non-self antigen, you will see, against microbial antigen. We are improving T cell functional persistence because we are targeting first memory T cells and that we are expanding a lot of memory T cells. We are able to block tumor escape mechanism. So simply because we are targeting several targets, for example, and that we are inducing a very fast and abundant T cell expansion that are able to tackle the tumor in an efficient way. So the manufacturing and the cost and the complexity is very easy. This is off-the-shelf synthetic peptide, and we have a very good safety, meaning that we have no off-target. We do not induce cytokine storm, despite the fact that we have a very, very high immunity. So, how does it work? I will go deeper into the mechanisms that we are using. So, as I said before, in the gut, you will find a lot of T cells that are almost memory T cells that could be here to protect the gut against bacterial invasion. Then you have CD8 T cells that are here, but you know also that you have a lot of T regulatory T cells that are here to induce a tolerogenic environment within the gut. So what we are doing is that we are mostly working on this pre-existing pool of CD8 cells that are here to check for the presence of invasion of bacteria within the bacterial wall, within the gut barrier. So, and we are working on molecular mimicry between bacterial antigen and tumor-associated antigen. Why? So, as you may know, there is a lot of publication of knowledge that highlights the link between cross-reactivity between bacterial antigen and tumor-associated antigen, but also against self-antigen. This is why it's very well documented that you have a lot of autoimmune diseases that are induced or that could be increased when you meet some bacterial species. So this is well documented for diabetes, but for also other diseases like cardiomyopathy, for example. What is also known is that when you are looking on some tumor-associated antigen and sometimes neoantigen that have been associated with very strong immune response in the patient, you will find some cross-reactivity between this antigen and some bacterial antigen, which is also a kind of demonstration that when you have similarity between non-self antigen and self antigen, you may be able to exacerbate a very high immune response against the self antigen. And then there is a lot of publication now that show that when you have a strong immune response, for example, when you are using immune checkpoint therapy and that you are able to induce a pool of CD8 T cells specific for TA or tumor specific antigen, there is a link between the quality of the response and the ability to find bacterial peptide or bacterial similarity with the tumor-associated antigen or neoantigens that are targeted. So this is exactly what we are doing. So in another way, this is what you can see on this picture. What we are doing is that we are targeting tumor-associated antigen. And you know that this is very difficult to target this tumor-associated antigen simply because almost all of them have been expressed during the development and are self-antigen, and so you do not find within the circulation a specific T cell able to react strongly against this tumor-associated antigen. Otherwise, you will have plenty of autoimmune disease. So by chance, if you look to the left, as I said in the beginning of the presentation, there is a lot of antigens that are mined by the immune system within the gut. And from this antigen, you may have the chance to find some that are similar to tumor-associated antigen because of molecular mimicry. So we have a lot of tools that allow to identify bacterial peptide, able to induce CD8 activation, and able to also induce activity against tumor-associated antigen because of cross-reactivity between the bacterial peptide and the tumor-associated antigen. So this is the principle of this approach and how it works into the patient, because we have already several clinical trials running. So we are delivering in mixture of 4, 5, 6 different peptides, so bacterial peptide to the patient that will enhance its kind of booster of immune response that will increase the number of specific CD8 cells and strongly activate them in a way that they will be able, because of cross-reactivity, to target tumor-associated antigen and to kill tumoral cells. So all of this is based on a dedicated platform that allows identification of the oncominic. So how it works? So we start on the knowledge that we have on the microbial antigen and all the studies that we have been able to perform on 2 samples from patients from different indications. So once we have identified tumor-associated antigens that we want to target, we have a lot of in silico process that will allow to identify bacterial mimics for the selected tumor-associated entities. We are working on MHC affinity, stability, prevalence of the peptide, but also, of course, on a lot of rules linked to cross-reactivity. Then once we have selected this tumor-associated antigen and this molecular mimic, bacterial molecular mimic, we are using in vitro validation, so we have a lot of tools to check that what we have found in silico, for example, affinity of the peptide for different HLA, could be confirmed in vitro validation. And then we are using also a lot of in vivo validation tools, especially we are working on humanized mice that are expressing HLA2, for example, which is the main HLA that we are targeting. So in this mice, what we are doing, we are working on immunogenicity of the compound that we are selecting, but also on cross-reactivity against the tumor-associated antigen, which is the more important criteria for us, and the ability to generate, of course, sufficient activation of cells to elicit cytotoxic activity, meaning that we increase a large pool of cytotoxic CD8 cells. And at the end, and this is probably the most important part, what we have found in mice, we are validating that into humans. So meaning that we are using a lot of blood sample from healthy people, and we are checking that effectively with the bacterial peptides that we have been able to select, we will be able to identify into the blood sample of healthy individuals CD8 cells already there and able to recognize the bacterial peptide. So confirming that effectively you have sentinel CD8 cells that are pre-existing in the body. We are using also this model to see that after activation and expansion of these cells with the bacterial peptide, we are able to induce CD8 cells that cross-react with the tumor-associated antigen. So basically, at the end of this pipeline, we have been able to show what we want to see in clinical trials when we will immunize the patient several times with bacterial peptides. And here I will now exemplify 2 or 3 clinical trials that we are currently running. So the first one is EO2401, which was used for solid tumor, cold solid tumor, so very difficult to target with immunotherapy. And for the first indication that we were selecting, it was glioblastoma. So basically, we were selecting 3 different tumor-associated antigens that are well known for glioblastoma. So IL-13RA2, FOXM1, and VIR5, which is also well known as survivin. So all of these proteins are overexpressed within the glioblastoma. So this is what you can see in red in the small picture. They are not or very low expressed in the normal tissue, and they are all called the tumor driver antigen, meaning that their overexpression is associated with very poor prognosis. So once we have selected this tumor-associated antigen specific for this indication, we have identified and designed some oncomimics, so bacterial peptide able to cross-react with this tumor-associated antigen. And you see that the oncomimic peptides for this indication are called EO2317, 2318, and 2360. So this is the peptide that we are using for immunization of the patient. So what do we have when looking at the results? So this is, for example, here, when you look to the left first, the level of immune response that we have in the patient treated with our compound therapy. So basically, this simply highlights the number of spots that we have. The level of immune response that we are able to address with any spot. What you can see is that we get a very strong immune response for all the patients that we have been able to investigate to date. So in this picture, in the cohort of 35 patients, we are able to observe positivity of immune response in 33 out of 35 patients. And in all the patients, while we were able to observe immune response against the different oncomimics, we are also able to show immune response against the tumor-associated peptides, so the cross-reactivity. This is what we want to see. And at the right, you can see also pictures. This is tetramer analysis that we are making on the blood sample from the patient. So here, this is after in vitro amplification of the cells. But what you can see is that when we are staining the CD8 cells with the different oncomimics, so EO2316, 18, or 17, we are able to detect a large population of cells reacting against this specific oncomimic. And from this, we have also a population of cells that are effectively cross-reacting against the tumor-associated antigen that we are targeting. So this is exactly what we expect from this concept and after several immunization of the patient. So Coming back to the study, so what we have done is that until now we have treated around 100 patients. The treatment is with different injection of the peptide in the patient and is used with nivolumab to accelerate immune response within the body. And in some cohort regimen, we also have bevacizumab to avoid, let's say, edema in the brain that could be associated with high immune response that we are inducing within the patient. Coming back to the immune response, so what you can see here is also tetramer analysis, but just after ex vivo setting, meaning that we are taking the blood from the patient and we are checking the level of immune response, a specific number of CD8 cells that are able to react against the tumor-associated antigen, so in yellow, or the oncomimic that was used to prime the immune system in red. So you can see that the two curves, so the response that we have against the oncomimic are very close to the ones that we have against the tumor-associated antigen, showing that we have a very good cross-reactivity with the peptide. But the things that are the most important on this picture, so when you have the level of CD8 cells and the time after immunization of the patient, is first that we are able to induce strong level of immune response in the patient very fast. So we are able to detect specific CD8 cells just after the first visit, meaning 2 weeks after the first injection. Then we have a lot of patients for which one we have level of immune response which is very high. For example, when you are doing such clinical trial, trying to induce immune response in patients, it's very difficult to find patients for which one you have more than 1% of specific CD8 cells in the periphery. And what you can see here is that we have a lot of patients with this level that is easily reached. And we have also patients for which one we are reaching more than 10% and sometimes more than 30% of specific CD8 cells against the tumor-associated antigen. So this is very huge number of CD8 cells that we are inducing in the patient and very strong immune response at the end. And the third point is that now we are able to see maintenance of the immune response in some patients after 100 weeks, so basically 1 year, 2 years, sorry, after the beginning of the treatment. So here also we are very happy with that. The conclusion is that we are inducing a very strong and fast immune response, and that this immune response can be maintained over time. So what is also important on that is the link with some exhaustion markers. So a lot of people, when they are using immune therapy, are afraid about the possibility to have still immune response, yes, but basically that all the cells are exhausted. So this is something that we are checking. And for example, here you have the level of different markers that we are able to investigate in the cells that are targeting specifically tumor-associated antigens. So for example, we are looking at interferon gamma, CD107, TNF-alpha, IL-2. So all of that are associated with activity of cytotoxic CD8 cells. And you can see that even after 2 years of treatment, a large proportion of the cells are co-expressing several markers, meaning that we have a clear maintenance of activity of the immune cell within the body. And last, what we have been able to see also within this clinical trial is that we have had a cohort which is very interesting because we were able to treat the patient before resection of the tumor brain tissue. And we have been able to do that on 6 different patients. And what we have been able to see is that only after 20 days, we are able to increase strongly the number of CD8 counts that we are able to see within the biopsy on the patient. So this is exactly what you can see here before and after the beginning of the treatment, and you have a strong infiltration of CD8 cells. So this is linked to the fact that we are inducing very strong immune response, and then that the cells are able to infiltrate the tumor. So on the clinical data, so this is not really my part. I will simply tell you what we have been able to see in this clinical trial. So currently we have very promising results with data that have been presented at the SNO with an overall survival that is around 40 months, which is 4 months over what is known using the current treatment algorithm for this patient. We have a very good duration of objective response. So it really seems to work. At least I will say in 1/3 of the patients in this very difficult to treat disease. So we are very happy with this result. I will go very fast on another indication because this is important. We were working on solid tumor, but also on liquid tumor, and we are targeting, for example, B-cell malignancy. This is important for us because there is a lot of different applications, not only in the oncology field, if we are targeting the B-cell. So we have another cocktail for which one we have been able to develop oncomimic targeting classical B-cell marker, CD20, CD22, CD37, and BAF receptor. I will not enter into the details of the clinical trials because of time, but basically we are using EO2463, which is the oncomimic mixture, alone, then associated with lenalidomide, and then associated with lenalidomide and rituximab. What we have been able to see in this clinical trial, which is just starting, is that here also we are a generation of very high level of CD8 cells, similar to what we have observed in EO2401 treated patients. These cells are cross-reactive, are able to kill B cells that are expressing the B cell markers, so lymphoma B cells. And very importantly, the immune response is maintained upon lenalidomide and rituximab treatment. So here also just a picture showing the establishment of the immune response. So this is the level of CD8 cells that we are able to detect against the oncomimics or the tumor-associated antigen over the time after in vitro stimulation. And what is very interesting is that you see after 40 weeks of treatment, we have already a complete response. That could be associated, of course, with use also of rituximab and lenalidomide, but we have on this patient complete response. And you can see in follow-up time, meaning that the patients are not treated anymore. We are still 1 year after the complete response, which is very good. So here also we are able to induce strong immune response and sustainable immune response. We have first result on these clinical trials, and currently I will say simply that we have 50% of the patients that have complete response rate after 90 weeks of treatment with EO2463, lenalidomide, and rituximab, which is better than all of the treatment regimen already existing. So I think this is the end of the presentation. So the conclusion is that we have been able to develop a new approach, a new cancer vaccine that relies on the primary activation of a T-cell, non-self T-cell repertoire targeting initially bacterial antigen. And because of cross-reactivity, we are able to induce a strong immune response against self-antigen and to kill tumoral cells. This is mainly associated with the pre-existing pool of CD8 cells existing into the body, the enhanced capacity to migrate to lymph node and the tumor, the increased lifespan that we have seen for the CD8 cells that we have seen in different clinical trials, and the probable better resistance that we have against inhibition that we have in the tumor microenvironment. And at the end, I will simply show this slide showing the different clinical trials that we are running. So we have PO24, one in glioblastoma and adrenal tumor, which is also a very difficult to treat tumor. We have EO4010 that is aimed to target every well-known tumor type, I will say the colorectal tumor, but also non-small cell lung cancer, breast cancer. So here we are targeting the 5-FOXM1 and other tumor-associated antigens that are overexpressed during the cell cycle. And then we have EO2463 that are targeting B-cell antigens. And then I will stop there. And if there is any question, I will be very happy, to answer.
Thank you so much, Laurent, for that really interesting talk. I know that it's very promising to all of us to see that these trials are looking so promising for those really difficult to treat illnesses. So we've got some questions that have come in already, so I'm going to move straight in and we'll get to as many as we can. I'll just remind the audience that if you do want to send in a question, and then just pop it in the box on the right-hand side, and we'll get to it as soon as we can. So our first one for you then is, how quickly can you develop a new immunotherapy for a specific indication?
So the process is very fast. So regarding the CMC, we do not have so many difficulties. This is peptide that we are using simply with montanide, and identification of incommonics and validation of incommonics basically takes 1 year. Starting from in silico investigation and then after in vivo validation and ex vivo validation. So after 1 year, we generally have one cocktail that is efficient, and we have, let's say, 1 year of regulatory things that will lead to clinical testing at the end. So 2 years from starting of the project to the clinics.
Excellent. Thank you so much. I know that that's a very promising time frame from start to finish there. I've got a question that's come in from Yulia. She has said, what is the tetramer, and is the structure of the tetramer important for the treatment results?
Yes, of course. So, the tetramer, it's a tool that will be able to detect the specific TCR that will recognize the peptide that we are targeting. So we are engineering tetramer specific. So we are docking, let's say, peptide within tetramer, which is part of the MHC, to get the two tetramer. Some others are using multimer, hexamer, but we are using tetramer. And so these tools allow to bind specifically all the CD8 cells that are specific for the peptide that we are targeting. And of course, this is very important, and we have been able to develop tetramer for each of the targets that we want to address. So for all the clinical trials, this is for on tetramer. But to be honest, some of them could be found commercially, not for all the targets, of course, because there is targets that we are the only one to work on, I will say.
Lovely. Thank you so much for that, Laurent. I hope that answered your question for you, Yulia. Another question that has come in, we'll get through as many as we can here. Which targets do you consider potentially attractive at the moment for future therapies?
So we have a lot of hope from the B-cell therapies, to be honest, but we can target a lot of different tumor-associated antigens. So when compared to CAR T-cell, for example, that are mainly targeting antigens that are expressed on the membrane, we have the advantage that we can target all tumor-associated antigens that will be overexpressed within the tumoral cell. So it could be cytoplasmic, it could be at the surface of the cell. Of course, we need to target peptides that are specifically presented by the MHC, and we have also some tools to validate that the tumor-associated antigens that we are selecting are the good ones. And so apart B-cell marker that could be used for a lot of different indications as lymphoma, but also, for example, for autoimmune disease, if we want to target specific B-cell population, we are able to target a lot of different classical tumor-associated antigens, as for example, R2 or step 1 a lot. We have already patented a lot of immunotherapy for prostate cancer, for example. So all the classical tumor-associated antigens could be targeted, and we have a list of our own, I will say, validated tumor-associated antigens that are less well known, but have the same profile that classical tumor-associated antigen, meaning that this is oncogene driver, and that are overexpressed mainly in tumor cells and not in normal tissue, I will say. We are also investigating now the possibility to use oncomimics for neoantigen. You know, for example, that there is a lot of neoantigen which are that could be shared from one patient to another one. This is the case for TP53, for CARES, for PI3 kinase. But unfortunately, most of these neoantigens are not strongly immunogenic. So this is why a lot of people that have tried to induce immune response against this neoantigen have failed. So oncomimics is really a technology that allows to induce strong immune response by cross-reactivity against neoantigens that per themselves are low immune inducers. So this is also one part of the work that we are doing.
Excellent. Thank you so much. I can see that there's lots of applications that you're looking to for the future, which is very hopeful for some of our patients out there at the moment. Now, I can see that we've got some questions from people who are much more well versed in this area than me. So you've got some technical questions incoming, Laurent. The first one here has said the underlying principle of molecular mimicry has largely been described in the pathological context of autoimmune disease. But could you leverage this concept in reverse to treat forms of autoimmunity?
Yes. So two things. Effectively, this has been largely described for autoimmunity, and this is exactly what we want to do. We want to induce autoimmune response against tumoral cells. So basically, we are trying to reproduce what sometimes happens in the nature. Apart from that, we have been able, with all the knowledge that we have acquired during this year, to change the activity of different set of T cells. So, for example, here we are inducing CD8 cells, but we have also the ability to induce CD4 cells, and we have also the ability to modify the level of activation of CD8 cells with appropriate oncomimics, so bacterial antigen, to at the end lead to exhaustion. This is not what we want to see in the tumor field, I would say. But for autoimmunity, we have some programs starting when we are targeting specific autoimmune target just to decrease the level of immune activation because of molecular mimicry. This is, I will say, more difficult than targeting simple tumor-associated antigens, simply because you have to target several autoimmune target, I will say, so antigen associated with autoimmune response. So you have to clearly define them correctly, then to find peptides that will be able to shut down the immune response in a way that they will also lead to immunoregulatory context that will avoid autoimmunity. And for autoimmunity also, as I said before, we are targeting directed B cells associated with the suppression of harmful antibodies. So there is here also a lot of application.
Excellent. Thank you so much. I'm fearful that we have too many questions for time. I can see that we've got one here from Amini and one here from Melanie. I will quickly do a quick fire round for those for you, Laurent, and then I'm afraid we will have to stop the questions there. So a quick one from Amini is, what about the structure of the treatment? So what would that look like for patients in the future?
So currently we are using peptide with montanide. So this is very easy to administrate. There is low side effects. So you have some side effects at the site of administration sometimes, so immune response at the site of administration. But at the end, all the clinical trials were set up with multiple injections of the vaccine. So now we are quite sure that we do not need uh, so much treatment. So all of the treatment are subcutaneous, so very easy to administrate to the patient. And we really think that probably in the near future, we can treat the patient only every 2 months, maybe only 3 months, every 3 months. So this is not really invasive for the patient.
That's great news to hear. I know that invasiveness of cancer therapies in particular is a concern for a lot of people. So thank you so much for that. And our last one quickly from Melanie. Firstly, she said, thank you for the interesting talk. So some glowing feedback for you there. She has also said that you had few data points for the later time points in the first trial. And she'd like to know, is that because the patients dropped out or because the patients are not far enough into the study yet?
No, so this is a very good point. So of course, all of the patients that we were able to do not have a very long follow-up because this is a very devastating disease. So basically, we were able to monitor a lot of people after 14 months, which is overall survival that we have currently. We have a third of patients that are able to go upon 18 months, which is very nice also. But at the end, the number of patients for which one we have followed more than 2 years decrease, of course, because of the number of patients that are still living, unfortunately.
Lovely. Thank you so much for that answer. Hopefully that answered that for you, Melanie, as is the way, unfortunately, with these clinical trials. But all data is fantastic data, and I can see that you're making huge strides in this sector, which I think is very promising for a lot of patients and family members that have been affected by these disorders. Unfortunately, we are very much out of time now. So thank you so much, Laurent.
Thank
Any
you.
questions that we didn't get time to get to today, do send them in still. And if you're watching on demand, please do send them in as well. And we will forward those on to the speaker as well. And as with all of the other talks in this event, this talk will now be available whenever you want to view it for the next 30 days in this platform. And that just leaves me to say thank you so much again, Laurent, and we will be back soon with the next talk.
Thank you. Bye bye.